ये नहीं आता पता चली गया क्लास तो वो हमें कैसे Falling down, please pick it up, Mother Teresa. For a lovely charitable work. Bless it on top of that. Do that. Bless the pot. I'm not a priest. I'm not a priest. I'm not a nun. I didn't know it.
मेरे पास तो नहीं है बैग मत है मैं नहीं सकता भाई उसमें विद्या है विद्या भी नहीं लेना मेरे को बैग दे तेरे फोन पे विद्या तेरे को विद्या मिली नहीं इसको तो
you want a CFL tour? Yes.
He's also church boy.
I
Yeah.
think church boy.
Church boy.
Oh, my God.
What is your phone?
मेरे पास है।
मेरे टीम का है तू
Yes, I am Christian. I am so nice.
या तो तू स्क्रीन का डाला he's also your combination is good. She is both Christian and Muslim. Mixed breed. Hybrid.
So we were talking about the process of digestion. Again, we start with this, the mechanism of digestion. To start with the concept of digestion again.
I mean, not giving my phone. I don't have your phone. So I need my phone. I'll get with my phone and not giving me back.
Nice. Oh, you know how to talk also. हाँ यहाँ पे क्या चल रहा है काम
फोन लेके वापस नहीं कर
हो गया अभी बुक निकालो फोन तुम बाद में सेटल करो तुमको प्रैंक करना जब आता नहीं है तो मत किया करो
वापस दे दिया
लेके 2 मिनट में कौन देता है इसे
इतना मुश्किल से 3 राउंड
इसको तो 3 दिन तक नहीं देना चाहिए था
आपके डर की वजह से ध्यान नहीं
हाँ, ये ऐसे वाले चीज में तो मैं बोलती हूँ और रखो 7 दिन अपने पास
देना
ओके चलो हमें मिला हमें नहीं तो वो शांति से पढ़ेगा भी नहीं आयो नहीं सीखेगा चलेगा फोन नहीं मिलेगा 2 घंटे तो Now we talk about the process of digestion. What we were already talking about, we had already come across the first step. And what was the first step we had learned? The first step of digestion in the mouth. We had already come across. Now, is there any other step of digestion taking place in the mouth? No, that is the first step and the only step of digestion. And what was it? We had been talking about the process of digestion of the starch with the help of salivary amylase. With the help of salivary amylase and resulting in the formation of what? What was the product formed? The product formed was, what did we write? What was the product formed? Maltose. Salivary amylase, also known as styrene. And the product formed was maltose. The product formed was maltose. And earlier I had also mentioned that saliva is released by the salivary glands, 3 salivary glands. Where are those 3 pairs of salivary glands present in and around the mouth? And what were their names? The parotid, the submandibular, and the sublingual. In the mouth, what type of digestion takes place? Only carbohydrate digestion. In the mouth, only carbohydrate digestion takes place. At the same time, when we talk about what about the proteins, what about the fats? No, the process of protein fat digestion does not take place here. Esophagus was the next part of the alimentary canal, but we did not talk about the process of digestion at all in the alimentary canal. Why? Because the esophagus does not carry out any process of digestion. It is only meant for passage of the food function. Now, what is the second step? The second step we are going to write in the stomach. What would that be? First of all, we have to remember this one, pepsinogen. What happens to pepsinogen? Pepsinogen in the presence of hydrochloric acid gets converted to pepsin. Nobody will talk here. Concentrate. By the time I finish those 9 steps, you will be extremely confused. और आप जब बात कर रहे हो तो as it is you are not understanding what I am speaking. अभी फोन मिला तो अभी पढ़ो. Not talking. Looking at your friend and constantly your mouth is working. तो उसको क्या बोले? What is other way I say? Say what is it? Speaking. So now this we are talking about pepsinogen. Did we study this step already? What pepsinogen? Inactive form of the enzyme gets converted to pepsin. How? With the help of hydrochloric acid. Now, complex proteins in the presence of the enzyme pepsin will be converted to peptones. Will be converted to peptones and proteoses. Now, in your book, they have just mentioned the general terms. So, I'm also going by those general terms. When you see our volume also some additional few terms are there. You may use those terms in your answer, not a problem at all. So pepsin gets converted to peptones. Now what are peptones? What are proteins? Same like carbohydrate, if you remember I told you that if I talk about carbohydrate, starch it may be having 500, 600,000 units put together. And when I say maltose, meaning much smaller now, but still not ready to go to the blood. They have to be even more broken down. Same here, when I talk about the difference between proteins and peptones, what are you supposed to understand? Proteins are very complex, maybe again made up of thousands and thousands of amino acids together, and this is now having comparatively now lesser. These are now having comparatively lesser, maybe hundreds and hundreds together, but they are still long chains. They cannot go to the blood, meaning they have to be further broken down to simpler forms. Stomach, what was that juice in the stomach? Gastric juice. And gastric juice we already studied. Gastric juice provides acidic medium. It is always responsible for the acidic medium. Now, is there any other digestion step that we have to study in the stomach? Nothing. That is all. In the stomach, no other step of digestion takes place apart from this. Now, there is one more when we are talking about in the infants. That is only in the infants to understand. In the stomach of the infants, there is one more step of digestion. See the volume. Is it mentioned about renin? Then we talk about this. Otherwise, there's no need of talking about renin. Check. Renin, is it mentioned? Process of digestion. Renin is mentioned in the stomach. Stomach, where is it? Stomach is not mentioned. No, renin is not there. No, renin no need for you. So when we are talking about renin no need for you. So now when we are talking about the So now, when we are talking about the small intestine, now we move on to the third part, and that is the small intestine. So the food now that is passing down, let us talk about certain more words. The food which was there in the mouth, what is the food in the mouth known as the mouth? If you remember, I already told you that with the help of the teeth, the mouth very much helps in the process of chewing and breaking down the food and mixing out with the saliva. So, what is the food in the mouth known as? The mass of food in the mouth is known as the bolus. What is it? The bolus. So, what is bolus? The bolus would run down the esophagus. As it runs down the esophagus and now it has entered the stomach, what is now this bolus termed as, this is now known as the chyme. Is it now becoming more or less semi-fluid? Yes, it is now becoming semi-fluid and the fluid nature keeps on increasing. So this word is very, very important for you. This remains optional, but this word you will have to remember. What is bolus? The mass of food that we swallow down and it is moving towards the stomach. In the stomach, the nature of the food is what? In the stomach, the nature of the food is acidic. And how is the food now to be made alkaline? The food will be made alkaline by the pancreatic and the bile juice. The food is made alkaline by the bile juice and the pancreatic juice. So now we are going to talk about the bile and the pancreatic juice. So when we are talking about the bile juice, let us learn this first. The bile juice, what is the bile juice that we are talking about? The juice released from the liver. Let us talk about the liver as the digestive gland now here. We are talking about the process of digestion that takes place in the small intestine. Whoa! Just now the break got over, no? What were you doing? Now, when we are talking about the small intestine brings about certain steps of digestion. What is the first gland to remember that it releases the juices? The liver. Now, this liver has got certain cells. What is the name of those cells? Hepatic cells. Remember, hepatic cells only, and it releases certain amount of water. It releases bile salts and it releases bile pigments. The name of the cells and all, no need for you. And now that we are talking about what is the secretion by the liver cells, roughly water, bile salts and bile pigments. What is this? Collectively known as, this is known as the bile juice. So the bile juice is produced by the cells of the liver, I am telling you. The bile juice is produced by the cells of the liver. Now that the bile juice is produced by the cells of the liver, if you come across that the liver is producing the bile juice, but it is temporarily stored in this small gland known as the gallbladder, and then from the gallbladder it will enter into the duodenum. So this bile juice which is produced by the liver cells, how will it enter into the duodenum? Because we are now talking about the process of digestion in the small intestine. So the bile juice will follow this pathway. The bile juice first of all enters into the gallbladder. So what is the job of the gallbladder in this process? Remember, the gallbladder is only storing. Is the gallbladder producing the bile juice? No, just storing it for a short period of time. And then this will enter where? This will enter into the duodenum. What is duodenum? The first part of the small intestine. That is about the liver that I had been talking about. I have to also remember about the pancreas. What is the location of the pancreas? See, the pancreas is also on the other side of the duodenum. So this pancreas also produces the cells of the pancreas. What would they produce now? They would produce, yes, a large amount of water, certain type of salts, and variety of enzymes. Here, names of certain enzymes you have to remember. Which enzymes I am talking about? Enzymes like trypsin, enzymes like amylase, and enzymes like lipase. So we can say, since they are all from the pancreas, there is a word always attached to them. Before these words we say trypsin, pancreatic amylase, pancreatic lipase, that way also. Because rest in the intestine always you will come across any type of carbohydrate digesting enzyme is known as amylase. Then how do we differentiate in which part of the body? We would say pancreatic amylase, intestinal amylase, like that. Am I clear? So what am I talking about? Now that we look into the duodenum, which are the steps of digestion? To understand that, We also have to understand what is the liver producing and what is the pancreas producing. Pancreas produces, here we are writing down only 3 enzymes, actually produces many more. The pancreas produces many more. And here, did you come across any enzyme that I have written down? The bile juice has no enzyme, remember. And the most important component of the bile is the bile salts. We are going to come across what is bile salts and its function. So as of now, remember the bile salts playing a major role, and in the enzymes of the pancreas, enzymes play a major role. Yeah. Then what is this? So I write here pancreatic amylase. I may talk about this as pancreatic amylase. I may talk about this as pancreatic lipase, just to differentiate when I talk next about the intestinal enzymes. What will this comprise then? This will comprise the pancreatic juice. This will comprise the pancreatic juice. Now, you get a question there. How is the food made alkaline? How is the food made alkaline in the duodenum? How is the food made alkaline in the duodenum? And your answer is just based on this concept that when we are talking about how is the food made alkaline in the duodenum, this is what you are supposed to express. Describe. The food is made alkaline in the duodenum because the duodenum receives the bile juice from the liver. The bile juice has got such composition. It also receives the pancreatic juice from the pancreas, and the pancreatic juice has got such type of a composition. So you have to describe this part and describe this part. Is it understood? So how is the food now, which is coming from the stomach? Is the food in the stomach acidic? Yes. The food in the stomach is acidic. So now this acidic nature of the food has to get converted to alkaline nature. What are the 2 types of juices helping in? The bile and the pancreatic juice helping in converting the acidic food to alkaline. Now let us try understanding. After that, we are a little bit aware about the concept of the pancreatic and the bile juice. Now we try understanding that what is the step of digestion then in the small intestine, the duodenum first. The first step of digestion in the small intestine is the breakdown of complex fats. I am numbering this equation so that you are able to understand. This is the first, this is the second we talk about, and now this is the third that I talk about. Now the complex fats are broken down by the presence of bile salts in the bile juice. And this will give rise to smaller molecules of fats. This will give rise to smaller molecules of fat. Now, this particular process, remember, I am not talking about an enzyme here. I already mentioned liver does not produce any enzyme. It gives rise to bile salts. The bile juice has got bile salts, no enzyme. Again, I repeat. Now, what is this particular process known as? This particular process is known as emulsification of fat. This is a very important concept. You are asked for 2 marks separately. This. What is emulsification of fat? What should you write? You will give this equation and then describe the process of emulsification of fat takes place where? In the small intestine or specifically in the duodenum to mention. The process of emulsification takes place in the small intestine with the help of bile salts. What is the meaning of emulsification? Breakdown of the complex fats to smaller molecules of fats. Clear? So now we have talked about one step of digestion of the proteins also, carbohydrates and the fats also. But many more steps to again talk about. So here we have to continue that what is the product and how do they further break down. That is how we understand the process of digestion. Follow me. Now I have to understand that now back in the small intestine, what about the carbohydrate digesting enzymes of the small intestine? So which is the 4th step that we come across? The 4th step that we are going to learn would be the starch which at all remain back in the food, meaning the food has run down to the small intestine. Possible that there are, and it is always that the food which is run down to the small intestine still has adequate amount of starch. Now what will happen to that starch? The function of that is almost the same like the salivary amylase. What am I going to write down here? That it is being acted upon by the pancreatic amylase. What type of an enzyme is the pancreatic amylase? This will also break down the complex starch to maltose, almost similar what the salivary amylase did in the mouth. Almost similar to what the salivary amylase did in the mouth. Now, so that is the starch step. The fifth step, I start from here now. The fifth step will be an extension of this step. The protein is partially digested. Now from here what? From here what? So now I talk about that this peptones, this particular peptones will be broken down by which enzyme? Which is the protein digesting enzyme of the pancreas, this one. So this will be breaking down the peptones to peptides, still smaller, still simpler, I would say, still more simpler. Peptones getting converted to peptides. But are they ready for going into the blood? Not yet. They are still not ready to go into the blood. What is the next set of steps? Few more steps, not to worry. I already told you that just in 9 steps you should understand the process of digestion. 5. I talk about the 6th step now. What is the 6th step? We are talking about fats. This has to now go on. What about this partially digested fats now? Are you following what I'm speaking here? How are we proceeding with the steps? I told you step number 1, step number 2, step number 3. Step number 4 is not an extension of step number 1, but step number 4 is something like, if at all the starches are present in the intestine, then what answer is, or the product is almost the same. 5 is an extension of 2. Understanding, no? 5 is an extension of 2 and 6 is an extension of 3. Again, I talk about smaller molecules of fat. You understand this way, so you'll be able to remember this. Smaller molecules of fats. Now, these smaller molecules of fats will be again further acted upon by what type of enzymes? When we talk about these smaller molecules of fats that we come across, this smaller molecule of fats will be acted upon by the pancreatic lipase. What are lipases? Fat digesting enzymes. So they will be acted upon by the pancreatic lipase. Fat digesting enzymes produced by the pancreas. So this will be broken down to again some simpler fat molecules. Simpler fat molecules or globules. Actually, there are stage names here, but in CBSE and NCT, they don't talk about these steps. Had those steps been there, the names would have been there, it would have been much easier for you to remember. So we'll have to stick to the syllabus. Now next is number 7. 3 more steps. So we are talking about 3 bit more digested. So let me say, number 2, when I say peptones, proteins to peptones and peptones to peptides. Understanding, no? I talk about fats. What is the first step? Fats to smaller fats, and smaller fats are still getting converted to simpler fats. It is like that to understand. 7, now what I pick up? I will again pick up starch. Maltose. Let me just talk about maltose. Maltose will be broken down further by maltase. Will be broken down further by maltase to glucose. And glucose. Is the glucose the simplest form? Yes, glucose is the simplest form which can enter into the blood. Process of digestion is complete. Glucose is the simplest form of digestion. Similar like glucose, there are some more types of sugar. I may talk about galactose. What is galactose? Galactose is the type of sugar that is also present in the milk, but the complex form. Maltose is one I talk about. I'm sorry. Maltose is one I talk about. The milk sugar I will talk about is lactose. These are the three steps you will have to remember this way. Lactase, and this lactase will get converted to galactose and glucose. Now here I am just giving you two examples. What happens to the different types of carbohydrate? They will all get converted into glucose. Galactose is also the simplest form. Okay, that is about the carbohydrate. Let us now take about the fats. Till where? Peptides, partially digested. To complete the process of digestion, peptides has to be broken down further. Peptides has to be broken down further. Which enzyme? The intestinal enzymes, intestinal peptidase. These are known as intestinal peptidase. What will they get broken to? Amino acids. Are the amino acids the simplest form to be absorbed by the blood? Yes. The process of digestion of proteins over. Where? Almost in the ileum now. Last step to understand the process of digestion. What are we left with? Fats. We talk about the simpler fat molecules. The last step. We talk about the simpler fat molecules. Now this simpler fat molecules, they will be acted upon by intestinal enzymes. I already made it clear that the set of enzymes that we name as lipase and amylase are produced by the pancreas also and down there by the jejunum also. The enzymes produced by jejunum, we will mark it as intestinal lipase. The moment I write intestinal lipase, you understand, produced by the jejunum. Simpler fat molecules now has to be broken down to what? Fatty acids and glycerol. Fatty acids and glycerol. So that is the end of fat digestion. The entire process of digestion you will have to study this way. I want all of you to take down the 3 steps, the 9 steps. Every type of a food nutrient. Carbohydrate, how many steps you get to see here? For carbohydrate, 1, and then the 4th, yes, the 4th, and then we talk about the 7th. Can you see here? So in this 3 steps, we are summarizing carbohydrate digestion. You get a question, explain, explain the process of protein digestion in the human alimentary canal. Which are the steps you are explaining? Explain the steps of protein digestion in the alimentary canal. Which are the steps you are explaining? Second one. And then the 5th. And then the 8th. Are you following? Yes. And then the facts. So here you will not generally get a question in your paper, explain the process of digestion of all the 3 nutrients. You generally get only carbohydrate or only proteins or some specific steps and the enzymes are something like this. So this is what we are understanding as how the process of digestion happens. Then what is the simplest form of carbohydrate digestion? Glucose. What is the simplest form of protein digestion? Amino acids. And what is the simplest form of fatty acid? Fat digestion. Fatty acids and glycerol. What is emulsification? Breakdown of fats where? Breakdown of fats where? With the help of bile salts. Where? In the duodenum. In the first part of the small intestine, in the duodenum. Clear? This side, clear? What is the enzyme which breaks down proteins in the intestine? The pancreatic enzyme. Dekho. Which is that protein digesting enzyme which breaks down peptones to peptides in the small intestine? Trypsin. Trypsin. You are not concentrating. So when we are talking about trypsin, trypsin is converting partially digested proteins to peptides. And there, when I talk about this enzyme, peptidase is converting peptides to amino acids. All of you complete this? All done? Now, what is the fate of these components now? We had already talked about. What is the fate of these components? The glucose, the amino acids, and the fatty acids. So now, back again to understand the concept of the villi sesicos. We had already talked about this villi. What were the two special features of the villi? The villi has a single layer or a single line of epithelial cells. And what was the other feature we were talking about? It is having rich supply of blood vessels. So now talking about all these molecules, I talk about these are the fatty acid molecules, these are the glucose molecules, and then the different type of protein molecules. What should happen if I understand that the process of absorption, what should happen if the process of absorption has to take place? The process of absorption means these molecules should directly fall where? These molecules should directly fall into the bloodstream. The entire process by which these molecules enter the bloodstream, this is what we are talking about as absorption. What are the two special features of the villi? Let us write down. Mention the two special features of the villi. Mention the two special features of the villi that bring about absorption possible. Mention the two special features of the villi that enable the process. Mention the two special features. Mention the two special features of the villi that enable the process of absorption. That enable the process of absorption. Two features of villi that enable the process of absorption. Remember, absorption means simple diffusion that is taking place from all the different outer surface of the villus. What are the two special features? Number one, every single villus has a thin wall. Every single villus has a thin wall. Villus, V-I-L-L-U-S. Every single villus has a thin wall made of a single layer of epithelial cells. That's the reason they are so thin. A single layer of epithelial cells. EPITHELIAL. 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 Epithelial cells. Number two, they are richly supplied with villus, richly supplied with blood vessels. Richly supplied with blood vessels. Richly supplied with blood vessels. Now, once you are aware about the process of absorption and the digestion over, now next part, after the process of digestion is over, we come across that there is another process to understand, and that is what we are talking about as the process of digestion, assimilation. Okay, let me talk about assimilation. So now from this particular region, the absorption has happened. So what are some of the processes to summarize now? What are some of the processes to summarize now? I may talk about certain words. We started with a process known as ingestion, how the process of food is taken in by cutting, biting, etc. Ingestion. What next after ingestion? We talk about the process of digestion. We had already studied the process of digestion, elaborate. And after digestion, what now next? A process of diffusion of the nutrients into the blood by the way of the villus, and that is what we are talking about as absorption. But now that the blood has all the nutrients, the blood will be circulating and distributing the nutrients all over. What is that process known as? This particular process is known as assimilation. What is assimilation? Assimilation is the process of distribution of the nutrients by the process of circulation of the blood. From where does the blood pick up the nutrients? From the intestine after the process of digestion, and then distribute all over, and all the undigested waste to be thrown out by the way of the large intestine. And what do we call this as? This is known as not excretion but egestion. This is called egestion. We are going to talk about excretion again, elaborate later. But remember the sequence that we had studied just now: ingestion and then digestion, absorption, assimilation, and then egestion. So this is about the process of digestion, alimentary canal, the digestive glands. The digestive glands that we had studied. Take out your volume, look into certain questions there, try solving some. Look into those questions, go to page number 154. Okay, look into page number 156 now. 154, that's stomata question we have done already. 156. Question number 1. Did you see there? Very short answer type. Number 1. What do we actually mean when we say that the enzymes are specific in their action? Pick that question. What do we mean when we say that enzymes are specific in their action? What does that mean? Now, if you see this story, can I say that this part of the protein is digested by peptones, and can these peptones also be digested by the same pepsin? No. For proteins, it is pepsin, and for peptones, it is trypsin, and for peptide, it is something else. So, which is the substrate and which is the product? Now, I'm talking about that this is the substrate, and this will be acted upon only by one specific enzyme. So that is the reason. Otherwise, we would have had only one type of a protein digesting enzyme all over. In the stomach also, in the intestine also. No. We have got more than 20 different type of protein digesting enzymes, step by step, break them down. So what is the answer that you write? What do we actually mean when we say that the enzymes are specific in their action? Just write it down in your volume itself, just the keywords, and later you can elaborate on that point. Every type of a substrate, or nutrient, is broken down by a specific enzyme. Every type of a substrate or a nutrient is broken down by a specific enzyme. For example, now you can pick up and take any two examples from out of these 9 to write. The easier ones to remember is about the proteins. The easiest one to remember, that protein breaking down to peptones by one and peptones to peptides by another. If there is space, you may just compare these two and write also. Or remember the step, I can write down step 2 and step 5 to compare and show, justify there for that answer there. What is the next question? Let me see there. Define photosynthesis. That you know. Define photosynthesis. What will you write? Biochemical process by which? David. एक बार फोन अगर मैंने ले लिया ना, पूरा साल भर नहीं मिलने वाला है. You will not talk. You are coming here to study. Somebody asking you the eraser. If I would have been in your place, what I would have done? Straight away give it to him. My focus should be on the teacher, what the teacher is telling. Your parents have sent you here for this. Your parents have not sent you here to do all the social service, give the eraser, give the this, and etc. In that case, the children who are not attentive in my class, straight away I will ask your parent, let them sit at home. No need to come here. You are not coming here to make friends, I'm telling you. You are coming here to get something above 95. That's it, right? Then coming here and doing all the nonsense. Define photosynthesis. What is it? See the condition. I finished with nutrition. I'm going to move on to respiration now. Photosynthesis का definition नहीं आ रहा है. Photosynthesis का definition shame. Really shameful now. What is the definition? Don't, especially boys, I'm again telling you. I see any type of a nonsense in the classroom, I will straight away call your parents from the office. I don't want this child in the classroom. Because later you end up getting 50s and 60s and it's a disgrace for us. Disgrace for us. I don't want my children to get 50 and 60. I'm not teaching for that. If you're not eligible to get 19 above, don't come and sit here. Straight away. You are sitting here again, I tell you, every child cannot afford this place. You are lucky enough that your parents can afford this. So give that worth back to your parents. Constantly I am observing you. Sometimes this, sometimes that. And even that. You are not disturbing, but you are not serious. You are not disturbing, but you are not serious. Be energetic children. Come to the class with lots of energy to learn. Lots of energy to learn. What is photosynthesis? तू बोलेगा तो तू बच गया, नहीं तो फिर से और सुनने वाला है. What is photosynthesis? I'll come to you. What is photosynthesis? Did you understand? Did you understand what is photosynthesis? फिर से बोलो. By which plants use their food and utilizing. रुको. Utilizing sunlight. The byproduct of the. नहीं. By utilizing raw materials क्या था? Utilizing water and by utilizing carbon dioxide, water in the presence of sunlight to give rise to glucose, oxygen and water. To give rise to glucose, oxygen and water. Liquid definition. You all are okay with the definition? Photosynthesis, boys will write down the definition. I'm not confident. The moment I see that you all are behaving like this, simple answers, immediately I'm going to call your parents. Immediately I'm going to call your parents. I don't want those thing in the PTM later that mark nahi mil raha hai. I will just make you sit in the at home. Sit and study at home. No need to come here. What is the definition of photosynthesis? Right. Right, and in 1 minute I'm going to ask you again. Let's see what are the other questions. Some few we try. Nutrition.
Mom, I had a doubt. You said with the definition of photosynthesis, biochemical process, and then again said biological.
नहीं, biochemical. Biological तो इन लोगों ने invent किया. Biological नहीं, everything is biological. Biochemical process. It is a What is it? Biochemical means it is a process happening in organisms with the help of chemical substances. Organisms is happening in the living organisms. With the help. Right. I'm going to ask you in a minute. What is the definition of photosynthesis? You will mention the raw materials. You will mention the usage of sunlight. You will mention the products. consume water. No, whatever food we are eating, that has got small amount of water. Now we talk about the roti and the different type of vegetables and the rice. How much of the water is there? The water content is so less. If we are not consuming adequate amount of water apart from that, then the movement of the food will not happen properly. If you don't take adequate amount of food, it is not only about the movement. The enzymes which are released are all in the concentrated form. And if the enzymes are not getting dissolved, they will not spread over the food. So to dissolve the enzymes, to make the blood also, if the blood is thick, how the distribution? The blood weight, the blood also has to be in the fluid form every time to bring about the process of distribution much faster. So the water is not only to drain, the water has got so many other functions, maintain temperature, etc. etc. Nico,
You all have the AC remote.
which class is this? Ninth, no? हाँ बोलो.
Biochemical processing which plants make the output by collecting carbon dioxide, water, sunlight, and fuel from the soil to give out glucose, oxygen, and water.
Super. अभी देखो ये अभी भी कॉपी कर रहा है.
कॉपी नहीं मेरा ही आंसर में नोटबुक में लिख रहा हूँ राइटिंग.
You will become very serious, हाँ. First exam में अगर खराब मार्क्स अगर मिला, I'm going to call your parent. First exam में you get odd marks, I'm going to call your parent. Now look into a few more questions there. Nutrition, move to page 157. Move to page 157. Number 15. Can you see short answer question number 15? Very fast. Let us just discuss some few questions. Number 15. Some questions I'm going to give you as homework. You will have to complete the homework. You don't complete the homework, I'll make you sit in the office and do the job. In one full week time, you can devote at least half an hour for the subject. Yes or no? Yes or no? Yes. See number 15. Explain the ways in which. No, this is number 6. Number 14 and 15 is not from nutrition. Huh? Number 16. Explain the nutrition process in amoeba. What is explain? They are saying. So explain means with the diagram you know now. What is the process name? Phagocytosis. Remain like this till the end. Huh? Now, the process name is phagocytosis. You may note down the process name is phagocytosis. That is number 15 I am talking about. No, number 15. 16. Number 16. Number 16 is phagocytosis. Next is what we move into.
In this question you have to draw the diagram.
Explain. If it is a 2-mark, 3-mark question, explain. Then you have to draw the diagram and explain the stages. I have asked you name the process. Name agar hai to खाली one word it is. Here it is explain. See there, it's a 3-mark question with the diagram. You have to explain. Number 17. Explain an activity with diagram to show that carbon dioxide is essential for photosynthesis. Activity to show that carbon dioxide is essential for photosynthesis. This activity we have done.
Yes, ma'am, we have done.
We did.
Which one?
We did only cover the leaf one and we started with the night.
No, that is actually presence of stars. I will tell you, this activity is a little bit different. Just let us take 2 more minutes. 2 more minutes. Now, now this particular question which is asked here, this particular question, they are saying explain the process activity with diagram to show that carbon dioxide is necessary for photosynthesis. Now, what are you doing here is carbon dioxide is necessary for photosynthesis. We generally take a potted plant. A potted plant is there, and then this potted plant we put it in such a way that there has to be a chemical substance known as, that is not, I think exactly that figure is not there in your book. That isn't there. Show me that figure. Show me that figure.
Even the solution reason it is there.
Correct. This figure. Check which page number 154. 154. So it is there, no?
Yes.
So this figure you will have to roughly draw this figure and explain. So how do you justify that carbon dioxide is necessary? The jar where KOH is taken and other jar no KOH, then what is the difference? We keep them for overnight or for a period of some 24 hours, 48 hours we keep. After that we take some leaves from here and go for the starch test, iodine test. If it is a plant like this which had KOH surrounding, the KOH will absorb all the carbon dioxide what was trapped. So these leaves, are they getting carbon dioxide? Not at all. Will they show photosynthesis? Not at all. Will there be starch? No. After iodine test, the leaf should appear.
If there is starch,
इसमें अगर photosynthesis नहीं है तो starch भी नहीं होगा. And now that I go for iodine test, what will be the color of the leaf? Brown. और इस दूसरे में जहाँ पे KOH नहीं रखा था, now they will show blue black color. क्यों blue black color? क्योंकि इसमें अभी starch है and iodine के साथ जब टेस्ट करेंगे always it will show blue black. आया समझ में? इसको टिक करके रखो. This is important with that reference. Now, what will you write in your notebook? Check. What will you write in your notebook? Just few more questions. Which one? Which page number we were doing? 157, right? Now, which page number 157? Now, see there, what is the role of hydrochloric acid? I don't want you to write again. Check number 20, number A and B. You know what are these? Number 19 we will do later. Number 18, energy requirement for fulfilled by which form unused copy. Now, try answering this question. Number 18. Explain the process by which energy requirement of the autotrophic organism are fulfilled. Correct. In which form the unused carbohydrates get stored? Starch. So your homework is write down. You will note down. कोई भी homework बिना किए नहीं आएगा. Number 18 you will write down in your notebook. Along with one question, explain process of protein digestion. Write in pencil, फटाफट लिखो. Explain process of protein digestion and this particular question to write down. Complete as homework. Only two questions. Explain the process of protein digestion and this particular question that I marked there.